and slides and the webinar itself will be made available to you at a later point.
Hi, this is Michael Oshinsky. I'm the Program Director for Pain and Migraine at NINDS. And today we're going to be doing a webinar on several funding announcements as a part of the HEAL initiative, specifically the discovery and validation of novel targets for safe and effective pain treatment. Okay, so that's the focus of the webinar today. NIH has been doing a large investment in pain research over the years. Before the HEAL initiative in 2017, NIH invested $516 million in pain research, and that's distributed among nearly all of NIH's institutes and centers. There are 27 institutes and centers at NIH. You can see the pie chart on the right where NINDS has the largest share of that, but not by much, the 13% of that $516 million. But there are many other institutes that really have a significant pain portfolio. In April of this year, Congress allocated $500 million to deal with the opioid crisis. That's dubbed the HEAL initiative, helping to end addiction long term. And $250 million of that has been reserved for pain research. What we're going to discuss in the webinar today is several of the funding announcements that have come out of this HEAL initiative that will provide the research community with an opportunity to contribute to accelerating the development of new therapeutics for the treatment of pain. As I mentioned earlier, the HEAL initiative is broken up into two components. This slide summarizes some of the goals, the key goals of the pain portion of the HEAL initiative. One of them is to understand the biological underpinnings of chronic pain. The second, the reason why it's highlighted, is because the funding announcements for this webinar really fall squarely in this goal of the HEAL initiative, to accelerate the discovery and preclinical development of non-addictive pain treatments. And again, it's only a part of that goal that's specific for these funding announcements. We'll go into that in more detail later. To advance new non-addictive pain treatments through a clinical pipeline, and to inform the best practices for effective pain management while minimizing the risk of addiction. If you want to learn more about the HEAL initiative, in the green box here you can see the link that goes to the NIH HEAL initiative website, where there's really just a tremendous amount of information. And at this point, there's more than 13 funding announcements that have been listed there, and that also includes notices of intent to publish, and for just pain alone. And there is also a JAMA article that was authored by Francis Collins, Walter Koroshetz, and Nora Volkov in the June issue that gives you lots of details of the HEAL initiative and its goals. So I mentioned there's a second half of the HEAL initiative, which we will not be focusing on here, but I wanted to address it in case questions come up. Here are some of the research priorities for the opioid use disorder and for reversing overdose initiative and research priorities. So one of them is to develop new treatments for opioid addiction. The other focus is to advance clinical trials for neonatal opioid withdrawal syndrome, to enhance the NIDS currently standing clinical trials network, which will build linkages to primary care, et cetera, establish a justice community opioid innovation network, and to also to optimize treatments for opioid use disorder and for reversing overdose. Okay, so that's really the last I'm going to speak about opioid use disorder, and the rest of the today was going to be focused on pain. The HEAL initiative at this point really includes a very comprehensive suite of funding opportunities for developing and accelerating the development and discovering new therapeutics for treating pain. Pain in all of its manifestations, acute pain, chronic pain, inflammatory pain, et cetera, et cetera. Some of the initiatives that are listed here are in the continuum from initial discovery of these targets for the treatments or the therapeutics themselves or even basic biology through preclinical development and then clinical trials themselves. The Acute to Chronic Pain Signatures Initiative is through the Common Fund. I'm going to give details on many of these later, so I'm just going to list them now. The discovery and validation of novel targets for safe and effective pain treatment is the focus of today's webinar. The preclinical screening platform, it would be a platform of animal models that are available for companies or academics that have potential treatments that they'd like to screen in animal models, either small molecules, biologics, or devices, and that will be done through a contract with a site or multiple sites. Translating effective stimulation devices for pain treatment was cleared by our council, and there are a suite of funding announcements that will come out for that for optimizing stimulation technologies or device technologies, even injectables, let's say an implantable pump, et cetera, for treating pain. And then there's the biomarker program, which will be biomarkers and endpoints for pain indications. There is a webinar coming up soon in November for that, and I'll talk about that later and what the date is. There's a data asset sharing partnership in order to funnel assets into a clinical trials network for pain that's being developed. And there's also a back pain research consortium that's being headed by NIAMS. Then a pain effectiveness research trials network. And then a pragmatic and implementation studies for the management of pain, which is being run by NCCIH. So again, this is a whole suite of funding opportunities that are being available. So even though the webinar today is focused on discovery and validation of novel targets, if we don't want you to take your project and really try to take a round peg and stick it into a square hole. We want you to really choose the right opportunity. And I'll give you details on that later. There's two other additional things I want to point out on this slide. One of them is that because of the many programs that we're developing to spend this HEAL money and to accelerate the development of therapeutics for pain or for opioid use disorder, there are many jobs that are becoming available. So if you know somebody who's interested, you can reach out on this website. And then there's the Pain Consortium link for research funding opportunities, not only through the HEAL initiative, but also that other $500 million that we've been spending on pain. You can find out how to apply for that also over there. Okay, so today we're going to be focusing on the funding announcements which are listed in the lower left-hand corner of this slide. And that's this RFA NS 18-043, and then the RF NS 18-042, which are an R01 and an R21 version of this discovery and validate novel targets for safe and effective pain treatment. And then I'm going to give some mention to an administrative supplement that's for validating a target that the main focus of the grant was really discovery. And then if the applicant would like to add on validation studies, that's available also. So here are the three funding announcements that we have available. The numbers are in black on the lower right hand of each listing. And there's one for the R01 version of discovery and validation of novel targets, the R21 version, and then here's a supplement. The supplement is a notice, NOT-NS 18-073. In your browser, if you search using these RFA or NOT numbers, you should be able to get access to the full funding announcement, which will give you the most full, most complete information that's available. And I'm going to just summarize them here and then take questions at the end for these particular funding announcements. There are many participating institutes and centers at NIH for these initiatives. The institutes and the centers that are listed here will be the institutes and the centers that will be holding these grants. That means those individuals that, for instance, the National Cancer Institute, if it's a cancer pain study, will be the primary on it. But the funds for funding these proposals will come from the HEAL initiative. You'll apply for these initiatives the regular way that you do, but on your cover page, you'll note these RFAs and all the requirements associated with them through grants.gov. Okay? The programmatic contacts at each institute for these funding announcements are listed at the bottom of the funding announcements. Because there's so many here, it doesn't make sense to put it on a particular slide. But if you do that search in your browser that I suggested earlier, at the bottom of the funding announcements, if you just scroll down, it's the contact names for each institute for the specific disease specialty or the institute that's responsible for that pain disorder that your target is specific for. Again, the initiatives themselves are not disease specific. It's pain in general. All right? Again, acute pain, inflammatory pain, chronic pain, nerve injury pain, etc. So now let's focus on what these applications will look like and a bit of how they'll differ from other R01s or R21s you might have written in the past. The research goals, the research plan must be designed to discover and validate a novel target for pain treatment. That's required for these funding announcements. If you already have a target that's been discovered, the focus can be to validate that existing target. But really, if there's any discovery activity still available, it'd be better to put those in too, just to really shore up the science. The science is the focus of the application again. Those could be targets for small molecules, biologics, such as an antibody or a cell-based therapy, or for devices, such as a stimulator or stimulation of a peripheral nerve or the spinal cord, or even deep brain stimulators, or even a device for injecting multiple drugs. But the focus of the biology has to be on the discovery of the target, not the development of the therapeutic, not the development of the device. Okay, it's the validating and discovering how that target fits into our understanding of the pain biology, and then validating it for a site for treatment. That's the focus of the funding announcement and needs to be the focus of the project. In addition, at the bottom of this slide, you see that experiments that demonstrate a lack of abuse liability are important for this funding announcement also and are a part of the review criteria. So the research goals have to have a sound scientific rationale that's supported by preliminary data or reference to well-designed studies that have been published that demonstrate the feasibility for discovering a target or validating a target as a good target for treating pain or for developing therapeutics on some other funding announcement. Again, the development of the therapeutic itself will not be supported by this funding announcement. That will happen on one of those other suite of different HEAL initiatives. The projects must include a plan for rigorous validation of the target, and examples are using multiple animal models, reproducing the work in another lab. Yes, you can subcontract the work to be key experiments to be reproduced in another laboratory. And that does not have to be at your institution. It could be at a CRO or it could be at another academic institution. Or demonstrating that that target is present in human tissue. That might be in the skin, it might be in a target organ, or it might be in the DRGs. And if you can get access to CNS from humans, even there also. Now, the relevance for pain therapeutic development should be mentioned in your application. Now, it's not going to be a large part of the application, but it should be mentioned in there in order to have the reviewers understand how you see work happening in the future to further develop this target, to develop therapeutics that can make it up into the hands of the patient. So what should your specific aims page look like? What should your research design look like for these proposals? So the critical gap, again, I should mention that this slide was taken from a figure on the URL that's listed at the bottom from the NICHD website, which has some great resources for your application development in general, not just for this initiative. And I particularly like this figure because of the way it summarizes the specific aims page. The top of your specific aims page should identify a gap or a critical need. In this case, it's specifically a scientific gap. You should not start your specific aims page with the epidemiology of pain. This is going to be a completely pain expert study panel. You don't have to tell them the number of people that are dying each year in the United States because of the opioid epidemic, or the financial burden of pain on the U.S. healthcare system. Unless you're studying epidemiology, that's not important. What's important is the scientific gap, and that's what you should be identifying in your first few sentences of your specific aims page. And then very quickly identify what the objective of the work that you'll be doing in this proposal to identifying or to filling that gap or addressing that critical need. Again, the objective is for this proposal, not your 10 or 15-year objective of what you want to get done in your lab. The next is the central hypothesis for this proposal, which will take the science, or the explanation of the science you're going to do to the next step, to be something that's really testable by the work that you're going to summarize in the specific aims. The specific aims will summarize the work that you're going to be doing to identify and validate a target for the treatment of some pain condition or pain in general. And then at the end of your specific aims page, you should summarize quickly what the expected outcomes are. Not just say that this will now be a target. Really get down into the nitty-gritty of the science for how these expected outcomes, and they should be directly connectable to the objective that you have identified at the beginning of your specific aims page. I hope this summary that I've given you about how the flow of the specific aims page should go helps you in designing your study and applying it to the specific research goals of this initiative that we're describing today. So the R21 version of this initiative, or this particular funding announcement, has a slightly different goals than the R01. And you should know that this is pretty across the board with what the difference are, the goals are between R21s in general from R01s. R21s are not small R01s or something that you just think you can finish in 2 years. It's very different activities, and that comes out in how they're reviewed. So this program announcement for the R21 is intended to encourage new exploratory, they're a bit more exploratory, and developmental research projects to discover something that's really going to be revolutionary or not necessarily an incremental scientific gap, but something really higher risk, because the hypothesis is that transformative. So projects that assess the feasibility of a means to identify or validate a novel target is an R01 or R21 type of objective, and not an R01 specific activity. So the unique and innovative use of existing methodologies, which are higher risk to this area of basic biology, which could lead to the discovery, would be more of an R21 type project. And I can go through these. These studies may involve considerable risk, but may lead to a big breakthrough, so that potential big breakthrough is really going to be an important part of the discussion during the review of an R21 type of application. So the research objectives and examples of the studies that are not appropriate for the funding announcement are similar to the R01 version. They're listed in the funding announcements. I'm sure I have a slide of this also. The supplements funding opportunity that I mentioned at our beginning of the talk are for currently ongoing R21 and R1s only. Those are the only two mechanisms that will be supported. There are some small examples that are exceptions, such as R37s and R35s, but there's just such a small amount of them that it's not listed here, okay? But really R01s and R21s, not your UO1s, not your clinical trials, etc. So these administrative supplements, if you focus down here at the last line, are for grants that will be ending before 2020 or 2021. That end date is at least 2020, March 30, 2020 or later, or March 30, 2021 and later. And the reason why there's two dates is because these supplements will support up to 2 years of funding, and the budgets are relatively large for supplements. They're up to $250,000 a year for currently ongoing R01s that are focused on discovering a target. And these supplements are for adding validation studies. These are for adding validation studies to currently ongoing R01 and R21 activities. And you can ask for 1 year of funding if your grant will go beyond March 30, 2020, and you can ask for up to 2 years of funding if your grant will go beyond March 30, 2021. There are no exceptions to these dates. They have to do with the requirements for the supplements, and then your grant cannot be in no-cost extension during this time. They actually have to be during the funding period. The goal of these is to have a very strong description of a validation of the target so that it can be developed as a therapeutic, and you have to have very rigorous controls and statistical analyses, and your power analyses have to be done for the particular experiment. You don't do one power analysis for a whole research project where you're using 4 or 5 different techniques. The power analysis should be done for each experiment. Okay, so now let's go back to this is still a supplement. So the examples of types of activities for validation that will be supported on the supplement, and that is target validation using animal models, so adding models that weren't initially proposed in the initial grant, or validation using human tissues, as I mentioned earlier, DRGs or RPSCs, even techniques that you have to develop in order to prepare you to validate them, such as designing genetic probes, or the creation or verification of genetically modified animals, like knockouts using CRISPR, Cas9, or the preparation of PET ligands for use in animal models. That will also be supported. Examples of activities that are not supported on these supplements will be validation of targets that are anticipated to have abuse liability. Those will not be supported by this funding announcement. Development of assays or probes to support basic understandings of the disease, so basic science studies that are not directed towards target identification or validation would not be responsive, or development of the actual engineering of devices or surgical procedures or diagnostics or rehab strategies, developing of those strategies would not be responsive to this FOA. And then the clinical testing of candidate therapeutics or clinical studies are not responsive. So you might be thinking at this point, but my project is translational. I do have a small molecule I'd like to optimize. I do have something I'd like to test in a clinical population. NINDS has a whole host of translational programs, such as the NIH Blueprint Neurotherapeutics Program, Create Bio, or the Translational Neural Devices, if it's a device, in order to support these activities. So I don't want you to think that NIH doesn't want these type of proposals. It's just that these funding announcements that I'm mentioning now for the target discovery and validation will not support those activities, but there's lots of programs at NIH that will. As a part of the HEAL initiative, there's also a large study that's going to be supported by the Common Fund for looking at chronic pain signatures. And these are the RFAs that are listed here if you'd like to be a part of that. Lots of omics discovery, proteomics, genomics, etc. Clinical trials associated with tracking phenotypic and sensory changes and genotyping changes in the patients that happen before and after some intervention. That will happen through this acute to chronic pain signatures Common Fund program and not through the funding announcements that I've listed. NCATS is supporting, through their initiatives, development of human cell and tissue models for the central nervous system and the peripheral nervous system that could be used for screening compounds for drug discovery. So if you have activities based on those, go to the NCATS website /HEAL and you'll see a list of those. And then, as I mentioned earlier, NINDS is setting up a preclinical screening platform for pain, the PSPP, which will look at small molecules, biologics, devices, and even natural products in a platform that includes a whole host of different animal models that represent particular diseases or the basic biology of pain. And so if you're interested in having your compound screened in this way, this contract is just coming up and running soon. So you can reach out to me for that or to John Keene. Associated with that initiative, we are having a workshop at near the NIH campus in Bethesda, Maryland on Wednesday, January 30th and Thursday, January 31st. This is a free meeting, and it's going to be a discussion. It's not going to be a series of didactic presentations. It's going to be discussion of about 30 to 40 people of experts in this field around which models should be chosen, what characteristics should be measured in those models, and how to deal with rigor issues such as statistical analysis and presentation of the data. So if you're interested in that, you can attend that meeting. There's the link for registering for it there, that HTTPS URL, and the site is listed there also, and some of the organizers. If you have an idea for a biomarker or an endpoint for clinical trials or even for tracking clinical outcomes in a clinical setting, there's the NIH Biomarkers Initiative that's a part of the HEAL initiative that you can respond to 2 currently open RFAs for that. And there is a webinar that's going to be happening that will describe the biomarkers program. Just in the lower right-hand corner of the slide here, you can see the dates for that is November 14th and 15th, and it'll be in the Washington, D.C. area. And then this HEAL initiative is a rapidly evolving initiative from the NIH, and it includes lots of moving parts. So I strongly encourage that you bookmark this web page, which is the NIH HEAL initiative web page, and then constantly go back and look at this funding announcement tab, which listed all the open funding announcements. On the right side, you can click on this website and sign up for updates. So as the funding announcements become available, they'll send you emails. So we're about 30 minutes into this presentation. And at this point, I think the most important thing we can do is start addressing some of the questions that you've been sending through. So don't stop sending them in. Keep sending them in. And we'll try to get the vast majority of your questions answered today. And of course, if you have additional questions, you can reach out to us in the future. I'm going to restate that all of these slides will be sent out to all of the attendees of this meeting. And at some soon date afterwards, we'll be sending out there'll be a link on the NIH HEAL website for the entire presentation in this webinar, okay? You will get the slides. All right, so let's start looking at some of the questions.
Just as a reminder, please use the Q&A portion of WebEx. It's located on the right-hand corner. Don't use the chat box.
What is the HEAL initiative? The HEAL initiative, HEAL stands for Helping to End Addiction Long Term. This is an initiative that was created at NIH in response to money that's been allocated from the Congress in order to address the opioid epidemic from a scientific perspective. That's in order to support scientific studies for studying addiction, opioid use disorder, and reversing opioid overdose. And then the focus of this webinar has been the finding new non-opioid and non-addictive treatments for pain. See, another question? Okay, are SBIR grants and RFAs coming? The SBIR grants don't need an RFA. They'll be considered for the HEAL initiative if they meet the HEAL initiative criteria. There may be an RFA. It won't be an RFA. There may be a specific funding announcement for HEAL forthcoming, but we can get back to you on that. But at this point, you can reach out to the SBIR liaisons at particular institutes. With respect to our institute, it's Stephanie Fertig. You can just do SBIR and NINDS or SBIR and any particular institute, and you'll find the contact there. If you have a project that's coming from a small company that fits the SBIR criteria, and it's in the HEAL initiative goals, that you should reach out to the institute's representatives to see if your project fits. Okay. The next question, I'm interested in learning if there's a later submission date for that. Yes. So the first submission date is November 27th. The next submission date is in February 11th, 2019. And then there's a third one in July 11, 2019. But those later dates are only if there's money still left after the previous ones, that they'll be considered. Can small business apply for this funding opportunity? The answer is yes. What about a novel? Small businesses don't have to apply specifically for SBIRs. They can apply for nearly any of NIH's funding opportunities. Again, you can reach out to program officials at particular institutions in order to get the details of how you register your company as an entity that can receive NIH funds. Next question, what about a novel target that has potential to treat several types of pain? How do you determine which institute to contact? So many of these proposals do come to NINDS. So I'll say reach out to me if it's appropriate for a diabetic neuropathy and a cancer, a chemotherapy-induced neuropathy. So if it's neuropathies in general, those proposals usually come to NINDS. You reach out to me or the other program officials, and they'll guide you to which institute should be appropriate. That's our job. Next question. 50% match. Okay, so the 50% match was for the 2018 funding. For the 2019 funding, going forward, these rules have changed. So for SBIR-specific proposals and your question of which funds it will be spent on, I encourage you to reach out to SBIR program officials, and they'll help you out with that. At this point, there is no concentration of HEAL initiative on career development awards. No, the supplements that I described do not apply for R43s or R44s, only for R01s. Hi, I'm interested in learning if there's a later submission date than November 2. Yes, there is. They're listed on the funding announcement. The next one is in February 11th, 2019. What are the application deadlines? They're listed in the funding announcement. The first one is November 27th, and the second submission date is February 11th, 2019. Would HEAL initiative support chemotherapy-induced neuropathy? Yes. And those applications, if they're specific for chemotherapy-induced neuropathy, will go to NCI, and Anomera is the contact, the programmatic contact. Would HEAL initiate? Okay, next one. When is the submission date? Yep, I already listed those. How does the RFA? Yeah, so the other RFAs, I think. So those are for could you put that number up again? RFA 18-41 is for the biomarker initiative, not for the target discovery and validation. There'll be a webinar for that on, there's a NIH workshop describing that on November 14th and 15th, and you can read the funding announcement to see the different goals. That's for endpoints and biomarkers for pain, not for target discovery for therapeutic development, which is the focus of this initiative. I don't understand the closing. I don't understand that closing date.
Closing date for applications.
I don't understand closing dates for applications. If you want to resend it with more explanation of what do you mean. I mean, the due dates are what I listed earlier, November 27th, 2018, and February 11th, 2019, et cetera.
Close next.
I'm studying this. Okay, I'm studying a niche pain area where opioids are used in treating the condition but they are not effective. Would such a disorder fall into this funding mechanism? The targets could potentially have broad application. Yes. The answer to that question is yes. If you're discovering a new non-opioid target, doesn't matter what the pain condition is. Okay, looks like it will be only 8 to 10. So you're right. Listed in there is 8 to 10. It's because we don't know what the budgets of those particular R01s are going to be up front. Although the funding announcement only says about 10 of them will be funded, there's a good hint that many more than 10 will be funded. Maybe it's only 10 per round, but have multiple rounds of funding. We got more than we initially applied for, and that's why the original RFA was written with only 8 to 10. The limit is not to only 500 per year, but you have to ask if the budget's going to go more than 500 per year. You have to ask the particular institute for permission to submit an application with more than direct cost of 500 per year. When you say target, do you mean an example of a cell type or molecule? Is this initiative appropriate for sex differences targets? Yes. It could be a molecular target. It could be a cell target. It could be a nerve in the case of devices. Yes. The target has a very, very broad definition. Will there be separate HEAL study sections, or at this point, the plan is for SPS to review them. Depends on the side. So it's planned to be what's called a RUMP-SEP, which is adding a day or so onto SPS. So there will be a lot of disease expertise and pain development expertise in the study section. ESI status is not considered in these applications for these RFAs. No. The budget limit I dealt with earlier, it can exceed 500, but you need permission from a program official beforehand to submit that application if it's more than 500 per year. Yep. I already addressed the chemotherapy-induced neuropathy one. How much emphasis should we place on novel target validation? That is a very important part of the FOA, the funding announcement. The roots of this funding announcement are in a series of workshops that were done with more than 40 companies and academics visiting NIH last year. And what we heard resoundingly from the companies is that they are loath to grab onto a new target until it's been properly validated, because over 90% of the work that they try to reproduce from published new targets is not reproducible in their hands. So my goal in developing this funding announcement was to have the validation studies that would be done in other labs or in companies in order to make a decision for therapeutic development will be done in the initial labs that do the discovery and published alongside that target discovery to speed the adoption of these targets for developing therapeutics by the company. So that's my goal. So the answer is Validation is very, very important, and that's going to be emphasized to the reviewers, not like a regular R01. Is the quality of life evaluation interesting to HEAL initiative? It's interesting to the HEAL initiative, yes. I'm not sure how that would fit into the target identification and validation of this particular funding announcements that I described here, but into the other initiatives it might fit. Is the discovery of a chemical or pharmacological small molecule pros within the scope of the R21? Not if it's for therapeutics. No, therapeutics developments will go through the translational programs that I listed in one of the slides, not through the target identification and validation. Developing therapeutics would not be responsive. If it's in order to develop a probe to test it as a target for pain, that could be developed, but it wouldn't be the initial discovery and screens. It'd only be a small amount of screening. It can't be the majority of the project. There will be representatives who can speak to you about HEAL at the NIH booth in San Diego. Yes, I'll be there. So if you'd like to say hi, come on by. There'll be a pain consortium. The goal is to have a pain consortium banner. So if you see the pain consortium banner, somebody be near it, or otherwise just walk into the NIH booth and say, HEAL, I need to know about HEAL. And there'll be people with lots of information for you. Okay, I'm a spine pain management patient. Should I be asking my new doctor if he's aware of the HEAL research? These programs are not at the therapeutic development stage yet. So I would say your doctor, the information from the scientific studies the scientific studies haven't generated information which would be useful by your physician yet in order to guide their treatment strategy. So I would say not yet.
Can you read the full question for them?
Sure. Yeah, I'm sorry.
Yeah.
I'm just trying to do this fast because there's so many questions coming through. I'm reading the question now. How do the RFAs you mentioned differ from the IGNITE grants for non-addictive therapies? IGNITE grants are for developing tools for developing a therapeutic. This is for discovering a target, so it's much earlier than the IGNITE grants. When is the expected release date for the RFAs for HEAL clinical trials research? They're in the pipeline. I can't tell you the date. I don't know. I'm not primarily responsible for those. That would be Jeremy Brown, Jeremy Brown. He's the program official at NINDS that's responsible for the Clinical Trials Network. I addressed this earlier, but I'll announce it again. Will these applications go through the SPS, which is a standing review study section at NIH? And the answer is yes, they will go through SPS. At this point, that's the plan. What will be the makeup of the study sections? They're going to go through SPS. So if you want to see the current members of that, look at the standing members. I'm sure we'll have to bring in additional members just because of the number of applications. The budget limit for the R01s are $500 a year without permission, over $500 per year with permission, and the R21s are up to $275 over 2 years, $275,000. That was asking for the budget limits for R01s and R21s. The next question, for validation studies, which is preferable? The same model replicated in multiple species or multiple models conducted within a single species? I would say yes. They're both appropriate, and it's specific for your proposal, which is more appropriate, and that will be determined by review, not by program, which I am. So we'll have to wait for review to determine that. Randomized controlled trials for novel use of existing drugs for pain conditions would be funded. That's the question. The answer is not under these funding announcements. Those will be under the clinical trials networks, which are being developed. Those are going to be phase 2 clinical trial networks, and Jeremy Brown is the programmatic contact for that, and they are not on the street yet, nor are they developed. So stay tuned. How would we know whether our target, say a channel, is already known but has not been tested in injury disease models, is novel enough to be competitive via this mechanism? You'll put in an application, and the reviewers will determine that. You'll have to write a cogent argument for how important it is for the particular pain condition or pain conditions that you suggest, and that's how the review committee will determine its relevance and importance. Can the aim be specific for a specific area of the body? For instance, if I want to do research on pain in the calf muscle for patients with peripheral arterial disease, the answer is yes. Yes, that would be responsive. Is continuous submission eligible applications to these grant deadlines? That's all up to the scientific review officer, which will be Kate Bennett. She's the one putting together. Program does not make those decisions. Is a letter intent required? It's not required, but it's highly recommended in order to help Kate determine which expertise should be needed on the review panel, so I highly recommend you send in letters of intent. Will you fund non-opioid behavioral approaches? Behavioral approaches. If you can design a study where you're looking at the target for those behavioral approaches. For pain, for addiction, they'll have to be under a different funding announcement, not for this one. And ways to validate that target for the behavioral approaches. How clinical-like does the validation of the target need to be? I'm confused about validation at multiple institutions. Good question. The validation at multiple institutions is having the work reproduced, the exact same experiments somewhere else. That's one type of validation. But one type of validation is not sufficient for this. Other types of validation are needed, such as multiple models or making a knockout to show that this is important in the pain pathway in some way. Could you go back to that question? I want to see if I got all the nuances of that.
Yeah.
So clinical-like, I'm not really sure what you mean. It would be very helpful for one of those the validations to show that the target exists in humans or is upregulated in a clinical condition. It's by testing in tissue, but you can't do it in the patients themselves because clinical trials are not supported by these funding announcements. How much preliminary data is needed in animal models? That enough to make the reviewers convinced. Considering that a lot of work that was done in the past that failed to translate, but to human peripheral neuropathies, but some of it has translated. So I wouldn't throw out all behavioral models. There is a place for them. But you're right. Showing it in human tissue would be important too. Please answer my question. I have a spinal degeneration. No, that's not going to. I can't answer the clinical questions. Since NCCIH is an institute that is also involved in this funding initiative, are you interested in other types of non-allopathic therapies? Yes. NCCIH is one of the participating institutes, and the interventions that NCCIH typically supports, such as natural products or acupuncture, etc., would be responsive if the study is not of the therapeutic or the intervention. The study has to be of the target, discovering the target, and validating that target for pain. For the administrative supplements, 3 pages are allowed. Does this include biography? No, it does not. And it's my understanding they could be more than 3 pages, but I could be wrong about that. The way you'll apply for the supplement is under the regular supplement announcement, and in your cover letter you're going to reference that notice that I listed on the slide, just for a point of information. Maximum grant per application. You can have 1 grant per application because grant and application are interchangeable words. If we have a high throughput method for identifying new targets, the experiments for understanding the science around the target would be hard to know. That's correct. Development or validating that high throughput method would not be responsive to that funding announcement, I suggest you look at the IGNITE initiative, IGNITE, at NIH, at NINDS, which I listed on the previous slide. I see that. That's not the whole question. Would be hard to know ahead of time. Is validation then mainly testing in animal models, human tissues, et cetera? The answer is yes. High-throughput screening is usually for therapeutics, which is not responsive to this funding announcement. Can you further explain any budgetary concerns for the R01 administrative supplements? It is my understanding that this will need to be itemized budget and not modular. That is correct. We need a detailed budget plan. You have to say what you are spending on, the money on, and it has to be detailed, just like you'd write any other detailed budget. If you're writing personnel in, you have to describe what those personnel would be doing on this particular supplement, not on the parent grant. If you're using certain reagents or you're sending the tissue out to be processed, include a quote for those materials. That's a detailed budget. Would tool development for identifying new targets fall under these RFAs? The answer is no. Look at IGNITE. Is an informatics-based approach generally considered inappropriate for these RFAs? I don't have enough information about the project, so you got to talk about what kind of target you're looking to identify. So feel free to ask another question with more detail. Can you clarify what target means? Peripheral nerve could be a target. It could be a particular protein that's on the surface. It could be a protein that's soluble inside, that's floating inside the cytoplasm, or it could be an area of the brain for a device for deep brain stimulation. It's a pretty broad definition for the target. Why don't you just put a specific example in and ask if I think it's a target. Is the development of selective chemical probes for novel targets appropriate? It has to be, if it's for like a fishing expedition for things that could be used as what? If you just want to develop a screen that where you're going to be screening tens of thousands of molecules to see if they work in pain models, that would not work. It has to be a basic biology study of a target. Will HEAL application deadlines continue for several years or just through 2019? At this point, we for sure have money through 2019. 2020 and forward, we'll get information on when the budgets are developed for those years. What type of clinical validation studies are appropriate to support proposed animal experiments? Clinical studies are not appropriate. It has to be in tissues that have been removed from a patient or humans, because human studies are not appropriate. NIH has a very broad definition of what's considered a clinical trial. So the vast majority of human studies research will not fall into validation studies unless you're dealing with the excised tissue. Please, can you restate the study section? SPS, somatosensory and pain. Somatosensory and pain study section. That's so if you go to the CSR website, and in their little search box there for the study sections, if you'd like to see the components of it and what its emphasis is, just type in SPS. Or just type in pain there. There's only a few study sections that come up, and SPS will be one of them. Next question. Target validation needs selective compounds, only if the target is small molecules. Would this program support any medicinal chemistry in order to develop selective tool compounds for target validation? Only a small amount. It can't be the majority of the project. The majority of the project has to be the target itself, and this will only be one sort of validation. So a small amount of medicinal chemistry could fit. I have an ongoing R01 from NCI that the focus is on chemotherapy-induced peripheral neuropathy, especially paclitaxel. I would like to test it in another pain model other than chemotherapy. What should I start? So you could, if you wanted to validate, if the goal of the supplement was to validate that the target that you've identified as a target for neuropathies in general, that would be responsive to the supplement, and you could add other neuropathy models such as diabetic-induced neuropathy. For targets for a device study, is targeting a specific peripheral nerve enough scientific rigor? It depends on, scientific rigor has to do with, not with the target selection itself, but it has to do with the evidence that's used to validate that target of the peripheral nerve. So I would say it would be scientifically rigorous if there were well-designed published studies or well-designed preliminary data or well-designed studies that you propose to do to identify it as a target. I would like to study and show that a relatively new equipment could be used to treat chemotherapy-induced neuropathy. Does it fit here? No. If you're testing a particular therapeutic, that means a device that can be used to treat, that's not responsive to this RFA. That would be responsive to other RFAs in the HEAL initiative. Specific for devices, refer to the slides that I showed earlier that list the other parts of the HEAL initiative, or go to the HEAL initiative website. Is this grant more suitable if we've already or have a possible target and trying to validate? You could do it that way, but you could also add more study of that target in order to contribute to the validation. So yes, it's not more appropriate. It's the kind of projects we're looking for. You have a good hint, you want to do the work to show it is. Does a receptor and its selective antagonist count as an appropriate target? So the work that you will be doing on the project is understanding the receptor and its place in the pain biology. So it's really not the target itself and the molecule that defines whether or not it's responsive to the funding announcement, but the type of activities that you plan on to do in your scientific study. So if you're studying that target to validate it, then yes, it would be responsive. That's all the questions for now.
For now, you have the opportunity right now to jump to the head of the line. We're going to give it a couple minutes just to give people a chance to.
Can you clarify when spinal cord stimulation is focused on validation rather than device development? Yes. You will not be developing, doing the engineering of the devices. What you'll be, an example would be placement of the device and looking at what the target is in the spinal cord for that device, and then optimizing showing that validation through showing that electrophysiological studies of that target to show that it's affected by the device, or molecular changes or genetic changes in that target area that's associated with your stimulation that shows that it can be used as a pain target. I am doing an NIH study section now. Can I apply 2-week extension? That's up to the SRO. So you have to contact the SRO for SPS, Kate Bennett, and she's the gatekeeper for whether or not you can use continuous submission.
Do you want to just answer the lack of abuse liability of targets?
So there's a question.
What does that look like?
So what does demonstrating that something has a lack of abuse liability? An example of activities that can be done to show that something doesn't have abuse liability are the condition place preference tests that are used for demonstrating abuse liability, or self-administration studies. Will the animal self-administer the compound? Those are 2 examples.
But if you don't have if you're just discovering and you don't have a compound yet.
That's right. So in some situations, it might be if it's a brand new target that there's no developed targets for that, then that would be difficult. You could do a knockout, and then you could do you can do the condition place preference or something.
Okay.
You're right. Some of them you're going to have to be creative. I don't have all the ideas in my mind. That's for the researchers,
okay.
You, the research community, to demonstrate in some way that it is or isn't. To show that it's not a target that's in the pathways that we know are important for addiction, such as the nucleus accumbens, okay? Or dopamine dependence. We are working on near-infrared light to stimulate a specific pain target. Will that fit? Yes, as long as you're studying the target, not the infrared light itself as an intervention. Would a GWAS study be considered for funding as a gene and target discovery effort? No. We found a new signaling pathway for an existing target. We want to study the pathway for pain as well as addiction, but we need to screen to find a pathway-based ligands. If you're really only doing a screening for ligands, that would not be responsive. So you should look at other funding announcements. If you're only okay, so is the understanding the mechanism of action of a potential new target responsive? If the mechanism of action, the goal of it is to understand its place in the pain pathway and how that leads to, and that would be a discovery and a validation type activity. But you should add more validation activities on it. If it's only to look at mechanism of action, and that's the goal of the project, then that's only part of the story. You got to put it in the context of target identification and validation so that somebody else will be convinced that it should be used for a therapeutic development. What about pain-induced affective disorders? Can that be a target? No. It has to be focused on the pain part, not the affective disorder. I already answered that question. Would studying biology and structure to ultimately develop inhibitors of potential targets be appropriate? So would studying the biology and structure to ultimately develop inhibitors, only if it has to be in the context of studying the structure to validate it as a target. So that means you have to be doing studies of the pain pathway and not just the target itself. There are mechanisms at NINDS and at other institutes for doing the work that you suggest for translation. That means that would be more of an IGNITE activity than this. In addition to biochemical targets, whether mechanical or electrical targets are appropriate. Yes, mechanical and electrical targets for device development are appropriate. If you want to study the target and validate it, the answer is yes. How to address that a new target for pain treatment is not addictive. So if there is a small molecule developed that's an agonist for that target, for instance, will the animal self-administer it? Or there are condition-placed preference tests that will show the abuse liability of compounds. Also, if you're not an expert in the abuse liability literature, I suggest that you reach out for a collaborator at your institution or another institution who is an expert in that area to develop that part of the application. I would like to target a specific area of the brain to target with optogenetic technique. Does this funding apply? If you're studying the target, the answer is yes. If you're studying the optogenetic technique for interacting with the target, the answer is no. Will the grant be responsive to a study comparing differential expression of genes from human sensory neurons, healthy controls versus painful, to discover novel targets? No. You know, you really have to, it depends on how the study is really based. If it's in order to understand the difference between painful and non-painful, then it's not responsive, because that's not a therapy target identification study. That's to understand the biology study. And if you're only looking to understand the biology and not to identify a target for therapeutic development, the answer is no. It really depends on the activities. So I'm sorry I can't give you more information. I need a more specific question. I have a grant that is in transition during my change of institution. Will this pose an insurmountable problem for applying for the administrative supplement? Depends on when the grant ends. But on our side, this happens all the time, people changing institutions. So the answer is no, it's not an insurmountable problem for applying. Are applications on in vitro models responsive? If you're studying a target for pain, yes, and that's one of the validation activities, then yes, it would be responsive. Go back again. Yeah, so ask them, Claire and or Clark and Yonkers, ask a more specific question. Which affective disorder? If you want to study depression, the answer is no, it is not responsive. If you want to study a target for depression, it's not responsive. If you want to study a target for pain treatment, then yes, it is responsive. Optimization of a specific neuromodulation method, locations, parameter, mechanisms for treating pain, is it responsive? It would be responsive if you're studying the target, not the treatment modality itself. If you're not studying the device, you're actually studying the target and saying what's the effect of changing the parameters on a particular cell type in the spinal dorsal horn, then yes, it would be responsive.
Okay, we're going to give it one more minute to allow for people to ask a couple more questions. If we don't get any, then we will end. Like we said at the beginning of this webinar, all slides and the recording will be made available to everyone that registered for the call.
So there was a question earlier. The text is, shame on you. How could you validate your research if you do not interface with actual pain management patients? Clearly, your response shows that your research or experience does not include actual humans like myself. So my experience does, for 14 years I worked in the Jefferson Headache Center. I saw patients 2.5 days a week for several of those years in order to understand pain in the clinical population. And the HEAL initiative does include research that will be done on patients with pain and these treatments. This particular research activities are earlier than that. They're not, when you're discovering a target, you won't do that work in humans for a whole variety of reasons. But the HEAL initiative does include actual applied and pragmatic research that will be done in the clinic. Okay, so the answer is it will be done on the HEAL initiative, just not with this initiative. Okay, more questions. Would MedCam optimization such as CNS penetration and bioavailability be appropriate in order to validate the CNS target? The answer is no, that should be done under the Blueprint Neurotherapeutics Program, BPN, at NINDS. Just reach out for that and you can get those funded.
And there'll be a BPN webinar on November 13th, so we'll be starting to advertise that probably next week.
Can you explain an ESI consideration for an R21 and R01s? ESI at NINDS is never considered for R21 applications, only for R01s. If ESI is for RFAs, it's typically not a factor here. So if you want ESI consideration, apply to the non-HEAL initiatives. No, you won't keep. If these are RR1, so you'll lose your ESI status if you get an RO1 on this initiative. Okay, but these applications are not going to be percentiled, and the way the ESI consideration works at NINDS is that there's an additional consideration above the percentiling. And since these applications are not going to be percentiled, the ESI isn't a consideration. Next question. I was wondering if these initiatives would also include the use of assistive devices to prevent the onset of claudication pain that occurs in peripheral arterial disease. If the research study is in order to determine what the target is, could be used as a therapeutic to treat the pain in arterial disease, then the answer is yes. But if it's to study to the device itself, the answer is no. Are in vitro models appropriate? The answer is yes. You could use in vitro models. I'm very I was wondering if these initiatives will include the use of... Okay, we did that. All right, it looks like I have answered all the questions. If you have additional questions, feel free to reach out to me. And again, the slides will be shared with those who were on the webinar or registered for the webinar. And we're looking forward to you developing these applications and submitting them in.
All right. Thank you very much, everyone, for coming out to the webinar.
Thanks, and have a good day.